In this third part of our video series on mitochondrial heat production and temperature, I'm going to outline the findings that we've obtained when cells are subjected to different kinds of stress. We will look at metabolic stresses imposed by substrate availability, ATP transport and usage, the availability of alternative respiratory pathways, and then by externally applied heat or cold. However, first, I'll recap very briefly what we covered in part 2. We considered how a mitochondrially targeted thermosensitive dye, MTY, allowed us to extrapolate the internal temperature of mitochondria, and how the findings accord with those using ratiometric protein fluorescence, for which an improved set of tools is now available. We observed how the temperature drop revealed by MTY fluorescence increases as inhibitors are used that block further and further down the OX-phos system, and how ratiometric protein fluorescence gives almost the same readout. Let's start by revisiting that observation regarding the use of different OX-phos inhibitors. The main conclusion was that different mammalian cell lines behave similarly, implying a similar steady-state mitochondrial temperature, and thus a similar contribution of electron flow entering the chain at different positions in different cell types. However, looking at the data in finer detail and taking account of appropriate statistical analyses, we can see some nuance in this broad conclusion. For example, 2 independently derived immortalized mouse embryo fibroblast lines both show almost no difference in the temperature drop produced by inhibition of complex 1 or complex 3 of the respiratory chain. In fact, when both inhibitors are applied simultaneously, the drop in mitochondrial temperature is no different than from either alone. Thus, we are able to conclude that almost the entirety of the thermogenic electron flow through the OX-phos system comes from the oxidation of NADH at complex 1 and is passed on through complex 3, with only a negligible fraction coming from other dehydrogenases that supply complexes 3 or 4 directly, such as from fatty acid oxidation. Similarly, in human HEC and U2OS cells, there is very little difference in the temperature drop produced by inhibition of complex 3 or 4, indicating only a small amount of extra electron flow separately into complex 4 from the EVR1 and similar pathways, whereas in IMFs, this is a far from negligible contribution to temperature maintenance. Note that in all cells that we've tested, the inhibition of complex 5 always produces a greater temperature decrease than the inhibition of complex 4, which is the terminal mediator of electron flow to oxygen. So either there must be some mechanism for maintaining the electrochemical gradient that sustains ATP production, or complex 5 itself, at least in some cells, some mitochondria, or some phases of the cell cycle, actually generates some of the heat that maintains mitochondrial temperature, presumably by a reverse reaction ATP hydrolysis. Note that this also results in proton extrusion, creating a proton gradient that has to be dissipated by some other mechanism, such as metabolite transport. This back reaction of ATP synthase may in fact only operate when respiratory electron flow is inhibited, because we needed to inhibit it to detect this. It's now time to introduce a major tool that we've used in our work, the alternative oxidase AOX. This enzyme constitutes a bypass of complexes 3 and 4 of the respiratory chain, passing electrons from ubiquinol directly to oxygen without contributing to the proton gradient across the membrane that drives ATP synthesis. Thus, when it's active, AOX releases all of the energy from that exergonic reaction in the form of heat, and none as ATP. Note, however, that in cells endowed with both AOX and complex 3, AOX normally carries only a minute portion of the electron flow under normal physiological conditions. It only becomes metabolically active when complex 3 is unable to reoxidize ubiquinol to ubiquinone and the amount of the reduced carrier builds up beyond a certain threshold that is hardly ever reached except under conditions of complex 3 inhibition. The alternative oxidase is phylogenetically widespread, being present in plants, protists, fungi, and even many animal phyla. However, it is essentially absent from 3 evolutionary crown groups of metazoans, namely cephalopod mollusks, vertebrates, and insects. So to study the effects of this inherently thermogenic enzyme on mitochondrial temperature, we transferred a cDNA encoding AOX from the tunicate Siona into mammalian cells as well as Drosophila and eventually into mice, from which we were able to derive AOX-expressing immortalized embryonic fibroblasts. When treated with OXPHOS inhibitors, AOX-endowed IMFs respond differently from control IMFs. Initially, when treated with rotenone, an inhibitor of respiratory complex 1, they show the same temperature drop as control IMFs, consistent with the idea that AOX remains inactive if complex 3 is not inhibited, the cells remaining dependent on heat generation by complexes 3, 4, and 5 independently of effects at complex 1. But treatment of the cells with inhibitors of complexes 3, 4, or 5 produced a different outcome. The temperature drop seen in AOX-endowed cells treated with inhibitors of these enzyme complexes was much less than control cells, indicating that under conditions of complex 3, 4, or 5 inhibition, AOX, being now activated, does carry at least some of the electron flow to oxygen and does so thermogenically. Furthermore, over the next hour of continuous measurement, the mitochondria of AOX-expressing cells appear to warm up again to reach close to the starting MTY fluorescence value, regardless of with which inhibitor they were treated. Of course, we can't absolutely rule out that a proportion of this might be due to mitochondrial depolarization and resulting dilution of the dye released into the medium. But on balance, it supports the idea that metabolism can be remodeled in the presence of activated AOX to restore mitochondrial temperature. And this occurs even at the expense of mitochondrial ATP generation. The exact details and mechanisms by which metabolism is remodeled are of course yet to be determined. We were surprised to discover that mitochondrial temperature can be maintained regardless of changes of substrate. Cells grown overnight in a low glucose or galactose medium that enforces OXPHOS as the main pathway for ATP generation show the same dependence on complex 1 to maintain mitochondrial temperature as control cells grown in high glucose. Starving cells of substrate did produce a slight, if statistically significant, decrease in mitochondrial temperature of a couple of degrees, at least after 30 minutes or so. But this was prevented or restored by adding back glucose. Treating cells with boncretic acid, an inhibitor of the adenine nucleotide translocase that shuttles ADP into and ATP out of mitochondria, produce a transient slight rise in mitochondrial temperature of just a few degrees, which then return to the starting value after 30 minutes. Once again, this seems to indicate a subtle remodeling of metabolism to maintain both mitochondrial temperature and metabolic homeostasis. In a further series of experiments, we investigated the effects on mitochondrial temperature of bringing about a major decrease in ATP usage by blocking cytosolic protein synthesis, a process which uses at least 30% of the ATP produced in a growing cell culture when it's not inhibited. Both control and AOX-endowed cells showed only a very minor increase in mitochondrial temperature, which had returned to approximately the starting value after 4 or 5 hours of treatment. AOX-endowed cells were able to withstand the stress of this treatment for at least 18 hours, whereas control cells were mostly dead by this time point, although surviving cells maintained mitochondrial temperature close to control levels. Drilling down a bit deeper, we uncovered a remarkable difference between control cells and AOX-endowed cells in the mechanisms by which they maintain mitochondrial temperature when cytosolic protein synthesis is inhibited, based on studies with MTY and exposure to OXPHOS inhibitors at different times. AOX cells were far more dependent on complex 1 for temperature maintenance under these conditions than control cells. Conversely, control cells were far more dependent on complex 5. Our tentative interpretation is that a buildup of ATP inhibits standard respiration and leads to over-reduction of intermediate electron carriers, notably ubiquinol. This activates AOX when it's present, enabling respiration to proceed with much less ATP generation, but with AOX itself supplying sufficient heat to maintain mitochondrial temperature. In control cells, there's no such relief valve, so heat must instead be generated by ATP hydrolysis at complex 5. Thermal stress followed by oligomycin inhibition of OXPHOS produced a multiphasic response in extrapolated mitochondrial temperature. After an abrupt but minor decrease resulting from cold shock, mitochondrial temperature as measured by MTY fluorescence declined by less than in control cells, implying once again a remodeling of metabolism to activate thermogenic pathways independent of complex 5. Heat shock cells showed an even smaller decline in mitochondrial temperature after oligomycin treatment, followed by an apparent rewarming and then a final cooling. Whilst these trends are very hard to interpret without much further experimentation, they do indicate a tendency for cell metabolism to adjust to external stresses so as to maintain mitochondrial temperature within a narrow optimal range. In the next video in this series, I will start to consider the biological implications of elevated mitochondrial temperature, starting with my own lab's work on Drosophila over the past decade, covering spermatogenesis, midline closure during development, recovery from chill coma, and responses to nutritional stress during development. Environmental temperature stress is something experienced by almost all land-based organisms, and even some in an aquatic setting. Extreme weather and temperature fluctuations are becoming more and more common in these times, whether we're speaking of the effects of droughts, floods, ferocious heat waves, or freak springtime snowstorms. Once again, I'll close by thanking my funders, collaborators, and colleagues. The work on mitochondrial temperature in cells has been largely conducted by senior scientist Mugen Terzioglu, now working together with Tina Salminen. The work on IMEFs was made possible by Praveen Dandapani and Luca Giordano, who established immortalized mouse embryo fibroblast lines from AOX transgenic mice and controls, and the AOX transgenic mice had been created by Martin Seebohr. And the fluorescent probes that we've used were developed by Young-Tae Chang and Takaharu Nagai, with essential proving experiments done in the lab of Pierre Rustin. Thanks again also to those who generated the images used here for illustration.